Vinayak Vittal is a 6-year biochemistry PhD student at the University of Washington. His research focuses on how human proteins recycle and repair cells. Please give a warm welcome to Vinny Vittal.
Thank you for the introduction. Are you guys ready to hear more about proteins? Good. All right. So today I'm going to talk to you about recycling. Living in Seattle, I'm sure we're all very used to recycling. We do it every day. But today I'm going to talk to you about recycling on a much, much smaller scale. It's something that's going on in each one of our cells, and it's happening all the time. So as Stella and Shivani told you, the human body is made up of trillions of cells. And each of these cells has a specific function. Our reproductive cells are important for reproducing. Bone cells are important for structural integrity in our bodies. Our muscle cells are important for moving, and et cetera, et cetera. But inside each one of these cells, we have something called DNA. And as Shivani mentioned, and Stella mentioned, it's actually an instruction manual that tells each of our cells what to do. And the way that we end up having different types of cells is dependent on what parts of that instruction manual gets read. A big part of what our instruction manual encodes for are proteins. So I'm a structural biologist, and when I think of proteins, I think of shapes. And the shapes that define these proteins are what define their function. So that's kind of a big mantra in structural biology, that there's a structure-function relationship, and how a protein looks defines what it does in a cell. So as a structural biologist, we don't just have to look at squares and circles and whatever some of these other shapes are. We can actually solve the structures of these proteins and look at them at an atomic level. So what you can hopefully notice is that each one of these is very unique, and they have kind of compact structures made from what we call helices right here, and what we call sheets, which are these kind of lines going back and forth. So the way these proteins look define what they do in cells. So let's take a few examples of different proteins, how they look, and what they end up doing in cells. So this is a protein called pCNA, and you can see right away that it has this large hole in the middle of it. It just so happens that this protein actually sits around DNA in the cells and acts as a clamp, much like this ring clamp. So it kind of loosely sits around DNA and is able to move back and forth and helps in replication processes for DNA. Other proteins have different structures. This is a structure of the protein collagen. Collagen's found in our muscles and in our bones, and when I think of when I see collagen, is kind of like braided steel. It's a very strong structure, and it's used to support in our connective tissues. Other proteins are important for carrying things from one place to another. This is a protein called hemoglobin. It's found in our blood, and its job is to carry these oxygen molecules through our bloodstream and take them from one place to another. You can kind of think of this as a fishing net, where it's basically capturing oxygen and able to carry it from one spot to another. So when proteins come together, they perform cellular functions. So I'm going to show you a video of what you're looking at here is an embryo of a sea urchin. And in blue is DNA, and in yellow is a protein called microtubules. And what you're going to see in this video is that the protein called the microtubule actually helps in order to split this cell into two different cells. So you can see the DNA floating around, it comes together in the middle, and then the yellow protein ends up actually splitting that DNA into two different cells. And you can see that happening over and over again in these different cells. Proteins are also really important for transporting things from one place to another. So again, in this video, what you're looking at is a microtubule, and you can kind of think of it as a highway in the cell. This big blue thing is a vesicle, which carries the cargo that needs to be transported from one place to another. And this motor protein right here is responsible for moving that across the highway. So you can kind of see it's actually literally walking across this highway as it's moving this cargo from one place to another. And this is happening all the time in our cells. This isn't real, this is an artist rendering, but it's still pretty cool. So another thing that proteins do is transfer messages. So what you're looking at here is a neuron, and these purple things are called receptors. So what the receptors do are allow chemical signals to create an electronic pulse that goes across the neuron. And when I play this, you can see that the receptors open up, a signal gets sent, and then it gets transmitted across the neuron. It gets to the end, and then it's able to transfer that from one neuron to another. So proteins are kind of a gatekeeper in this signal process. So another really important thing that cells do is recycle. And this is what our lab studies. We're interested in how cells recycle and how the proteins involved in this process work. So the first question to ask is, who actually does the recycling? And it just so happens that there's thousands of proteins whose job in the cell is to act as recycling machines. My lab studies this specific family of proteins and how they work to recycle. So in our lives, we have different categories of things that need to be recycled. We have our metals, our plastics, our glass, and our papers. And each one of these has a specific machine that does the job. So for an aluminum can, we have this machine where the aluminum comes in and it spits out a recycled material. We have the same thing for something like plastic. It gets turned into a different machine and it comes out a recyclable material. But what we know is that we can't put the aluminum can in the plastic recycling machine and the plastic bottle in the aluminum recycling machine. The machines are made for specific types of recyclable materials. Well, in cells, the types of things that need to be recycled is exponentially larger. We can't use a Dr. Pepper recycling machine for a Coca-Cola recycling machine. Each one of those different types of sodas needs its own machine. So we really have a problem where we have many, many more recycling machines because we have many, many more things that need to be recycled. So maybe an even bigger question is why do cells even need to recycle? So for this, I'm going to take you to Naples, Italy. In the early 2000s, there was a trash strike in Naples, and basically no one was coming up to pick up the trash and there was no recycling. So what ended up happening was that the streets just began to become filled with trash. This really just became difficult to move from point A to point B. People couldn't walk. And eventually it started to become a real health hazard with all this trash building up in the city. So this is really pretty similar to what ends up happening in cells. So what you're looking at here again are neurons. So this is a picture of what a healthy neuron would look like. And you can see these nice endpoints going from one to another. In Alzheimer's, what ends up happening is that basically cellular trash starts to build up at the end of our neurons, and it really blocks the messages being able to be sent from one neuron to another, and this manifests as the disease that we know as Alzheimer's. So what is this trash that's building up over here? If there's one theme for this evening, it's going to be that it's proteins. So proteins are also the trash that actually ends up building up at these ends of neurons. So why does this happen? So just like us, proteins end up aging. So if we're looking at time going from here to here, in the beginning we have a nice folded healthy protein. It's compact, it has its structure, and it has its function. As time goes on, sometimes proteins start to get unfolded. So they start to unravel a little bit, and they don't have this well-defined structure that you see here. Eventually what can happen is when these unfolded proteins start to interact with one another and form these bundles. And these are what become toxic in cells. So that's kind of representative of what you're seeing here. Basically unfolded proteins that are coming together and forming these globular regions. So what's the solution to this problem? Well, the solution is to be able to clear these proteins in our cell before they get to that problem point where these large bundles. So the focus of recycling proteins is to get them early and to start recycling even healthy proteins and these partially unfolded proteins. So how does it work? So this is our cell. This is one of our recycling proteins that we work on in our lab that I showed you. And this is a piece of trash that is not real in the cell. But what it actually would be is a protein. So our recycling protein would start floating around until it found the thing that it needed to recycle. And it's kind of that lock and key interaction that Shivani was talking about. It's then able to carry it and take it to this recycling machine. The recycling machine breaks it down into its chemical parts that can now be built up back into this original protein. Now say the same machine came across a different piece of trash like a plastic bottle, but in this case it's this green protein. It'd go around in the cell, but this particular piece of trash doesn't fit with our recycling machine. We need a different one that does form that lock and key interaction so it can bind. It'll grab it, take it to this machine, break it down, and build it back up. So the big questions that we ask in our lab are, what do these new recycling proteins, the machines that are doing the recycling, look like? And what do they target to be recycled? So what are the pieces of trash that they're looking for? My particular research was to study one of these recycling machines and to understand what its respective trash was that it was looking for in cells. So like I told you, my lab studies this particular family of recycling machines. And although we know what a lot of them look like, we don't know what all of them look like. So when I came into the lab, my job was to study a particular machine that we didn't yet have the structure for. So this slide is 2 years of work that is now one slide, but it's okay. It's for a good cause. So this is the protein that I solved the structure of, and what you can see is that it's a little bit different from the other ones, and mostly in that it's very flexible and then it's moving. When you compare it to all the other ones, which are very static, you can see that this one stands out and has this unique role where it can actually move part of its structure. So like I told you, the big important question is, how does the structure and function relate? What we started to find out was that my particular protein liked to recycle these partially unfolded proteins and not these healthy proteins. So we kind of narrowed it down a little bit in terms of what its trash was. But before I can tell you more, I'm going to explain a little bit more about unfolded proteins. First of all, unfolded proteins have a starting point and they have an ending point. And there's different types of unfolded proteins. Sometimes the unfolded region can occur at the starting point of a protein. Sometimes it can happen in the middle of a protein in a large loop. And sometimes the unfolded region can happen at the end of the protein. What we started to learn is that my particular machine was very good at recycling proteins that had these unfolded regions at their start. To complicate matters, what we started to learn is that there's thousands of different proteins that have disorder at the start, and they all look a little bit different. So the big question is, how can my protein recognize all these different pieces of trash? That was the question. So how can it recognize all these different pieces of trash? And what we realized that it had to do with this flexibility that it was using. So let me give you an example back to our cell. So you can imagine that my protein's floating around. It's doing its thing. And it comes across this piece of trash that has this unfolded region at its start. And it's able to interact with that one because it has all these different modes in which it can interact. If it found another one, it would use a different confirmation of its flexibility in order to interact with that. And yet the same for another one. Eventually it would find a position that was good. It would lock in on that substrate. It would take it, drop it into the trash, break it down, And when it built it back up, it would try to it would be refolded again and not in that unfolded state. So the message that I want to get across to you is that recycling is important in both our real lives and in our cellular lives. It's a complicated process that involves many, many different proteins. And it has serious implications for human diseases, especially something like Alzheimer's. With that, I'd like to thank the Engage program. It's been a great experience. The class was amazing, and Shelley and Juliana were amazing instructors. I'd like to thank Town Hall Seattle for letting me come here and talk, and my department, the Department of Biochemistry, and my boss, Dr. Rachel Clevitt. That'll take your questions.
What kinds of cells is your recycling protein involved with?
All of them.
Oh, is it every human cell?
Every human cell has a recycling process.
Well, it has a recycling, but you studied a specific protein. That's
my specific one. Oh, it's known to be in neurons. It's known to be in developmental cells, and we think it's expressed in most cells, but it's overexpressed in neurons mostly. So.
I'm just curious. You say break them down. Break down the proteins.
Right.
Reassemble them completely. Bonds broken and whatever, or is it just?
Yeah, no, it really does. So proteins are made of these building blocks called amino acids, right? So the actual blue recycling machine will break it down into those very basic parts, and those will float in the cell and be built back up in protein making processes.
Chemical
Yes.
action versus physical.
Yes, exactly.
So I understand that your proteins, your trash collecting proteins, are specific to certain types
Yes.
of unfolded protein, to trash.
Mm-hmm.
But I noticed that the trash can is generic. Is the trash can generic, or is in fact selective to what proteins it processed?
It is actually generic. So the trash can, to say this again, it's actually a bunch of proteins that make this cylindrical structure in the cell. And you can feed in any sort of different protein and it'll break it down into its components.
Yeah, which is different from our recycle.
Yes, exactly.
Got it.
Yeah, so the carrying machines are kind of the unique ones.
Yeah.
The actual machine that breaks it down is very generic.
Okay. And the amino acids that result are just floated free then into and then are available for
Yes.
the protein replication.
Exactly.
Okay, thanks.
Mm-hmm.
After the trash goes into the recycler and everything comes out and it's reused, does it always go back to the exact way that it was before?
No, so I told you it's broken down, and so there's 20 amino acids that make every protein essentially. So I use it as an example that you could imagine it being built back up into itself. But those components could be used to build any other protein that the cell would need at that time.
So how does it decide on what the cell needs?
That's a really good question. The cell somehow knows what it needs when it knows that it needs it. And that's why we do the science that we do half the time, is to figure out why it's going on at the time that it is. So there's certain chemical signals that tell a cell to do what it needs to do at a certain time.
So we've heard a lot about the lock and key analogy,
Mm-hmm.
and it sounds like a large part of that is the structure of the proteins.
Mm-hmm.
And so my question is that when you start to get to an unfolded protein and the structure degrades and possibly degrades in an unpredictable way, how are you able to create a protein like your protein that's able to lock on to this kind of ambiguous structure?
Right, so the beauty is that the ambiguity in my protein, the fact that it is flexible, allows it to kind of recognize all these other flexible regions. So in terms of like the chemical bonding, the fact that it is flexible gives it like a very large platform in order to form interactions with these other things that are very flexible. So the lock and key is very ill-formed, and it's almost stochastic or by chance that some of these interactions occur.
So would you say that particular lock and key analogy is more of a chemical one as opposed to a structural one then?
For mine, yes, absolutely. Yeah.
Thank you very much. Excellent use of analogy to convey the different ideas that are involved. When you're talking about your carrier protein that is latching on and bringing it to the recycle mechanism, at that point, is it able to release, or is it also part of the recycle process at that stage?
The general answer is that those proteins are able to release. So they release and they're not degraded, and they move on and keep on doing their thing. But to be more specific, even the recycling proteins get recycled eventually.
So this recycling protein is not a protein that we generate with our body. It's like something that you come up with. And
No, it's in our bodies. Yeah, all of them are in our bodies.
We already have this. And
Yep.
We're looking for that. How are you going to say this protein go to where? What you were doing? I mean, that's so special. I mean, if we have it already.
Well, you're absolutely right. So what we're doing is basically basic discovery, right? The fact that we have these, we didn't know that until we started doing the research to get to that point, right? So I understand what you're saying, but yeah, it was basic. We didn't know about it until we started looking for it, essentially. So we're learning as we're going on, is basically what it comes down to.
If the recycling proteins doesn't do their job, they don't recycle, is there a punishment for them?
Yeah, I mean, part of it is disease, right? Alzheimer's is part of the punishment, I guess. You start to build up these what are called plaques, and that's part of it. Certain cancers, these recycling proteins get malfunction, and cells have uncontrolled growth at a certain point. So it's not just about manifesting as these large globs, but you need to be able to degrade proteins that are responsible for specific processes at the right time. Otherwise, cells become out of control, essentially.
So recycling requires a lot of energy. So is part of your research, or the direction of your research, eventually going to, instead of having to recycle, breaking down amino acids to then rebuild them to the protein structure, is there a way to repair the protein structure and refold the protein back to its original state, and by repairing instead of having to recycle?
So there are proteins in the cell that do that. They're called chaperones, and their job is to actually try and refold proteins at a certain time. So that process is going in a parallel fashion to this degradation process. So the cell does actually have kind of a refolding mechanism already, but sometimes it gets to a certain point where you just have to get rid of it before it gets too late.
So this is a stochastic process, this catching of the protein, because it's very complicated. So that would suggest that it misses sometimes, and that certain diseases like cancer are inevitable. Can you make any calculations as to what you would expect the incidence of missed bad proteins would be towards that?
So it's stochastic in a certain sense. A lot of the times, these specific machines are localized to specific regions in the cell. So their function is supposed to occur in that particular compartment. And that kind of lowers the stochasticity. In terms of a calculation for how often it misses, I unfortunately can't probably put a number on that. Mutations that occur in these machines would increase the risk of some of that type of thing happening, but in terms of general missing, I'm not really sure. Okay, thank you, appreciate it.